Welcome to the unit on microbial pathogenesis. This is one of my favorite ones to teach because this is what I actually study, with TB being my pathogen of choice. I actually teach the upper level microbial pathogenesis class here at UVU, so if you're interested, come look for me again and take that class with me. This video itself, we're just going to start with an overview about what microbial pathogenesis is and talk about some key terminology you need to understand how we study microbial pathogens. Let's go ahead and take a look. All organisms that share an environment are going to have some sort of relationship between those two species. There's three main types of these symbiotic relationships. The first is mutualism, and that's where both species benefit. So the majority of our healthy human microbiota have this type of relationship with us. They benefit by living in us or on us, and we give them a good habitat and food, and they give us things like the ability to digest certain molecules. They help our immune system, and we're learning more and more about the things that the microbiota do for us. The second type are commensalistic relationships. This is where one species benefits and the other really isn't affected, so they don't really care very much. We used to think that the human microbiome had this type of relationship before we started learning all the things that the microbiota actually do for us. And then the third is the one we're going to focus on throughout this unit, and that's parasitism. This is the relationship where one species of the pair benefits and the other one is harmed by that interaction. So microbial pathogens have a parasitic relationship with the human body. We're harmed while they benefit by living in or on us. So some key terminology from this section. The first is we're dealing with infectious diseases. So you have to understand that a disease is just any condition where the normal structures or normal functions of the body are damaged or impaired in some way. Pathogens themselves are microbes that cause a disease. And so they're microbes that have a parasitic relationship with us, and the diseases that pathogens are called infectious diseases. This is a little bit confusing because all pathogens have a parasitic relationship with us, but when you call a pathogen a parasite, in medical usage, this is typically reserved to diseases that are caused by protozoa or worms, so some of those eukaryotic pathogens we learned about. So while all pathogens have parasitic relationships with the human body, only eukaryotic microbes that are protozoa and worms are really called parasites directly. Then the last term is the pathogenicity, and that's really what we're going to study throughout these chapters, is the ability of a microorganism to cause disease. And so obviously not all microbes are pathogenic, and not all of them cause disease. The study of pathogenicity is trying to figure out why one microbe might cause disease while another one doesn't. Now, all diseases, including infectious diseases, are defined by their signs and symptoms. The difference between these things is that signs are something that is objective and measurable. So for example, if you're running a fever of 102, that's a sign of an infectious disease. But symptoms are subjective. They're typically the things that a patient would feel or experience. And so if they're feeling just kind of tired or achy, those are symptoms of a disease. When you look at infectious diseases, they can be communicable or non-communicable. So what that means is that communicable infectious diseases are contagious. They're spread from person to person. While most people think of all infectious diseases this way, they actually aren't. There are some infectious diseases that are caused by a pathogen, so a microbe coming into the body, but that can't be spread from person to person. A good example of this is tetanus. So you can get tetanus if you step on a rusty nail and you introduce the tetanus bacteria into the body, but you don't actually transmit that to anyone else. So that'd be an example of a non-communicable infectious disease. And when you look at infectious diseases, an acute infection, so one that has a short time frame, has characteristic stages that we can see here. The first is the incubation phase. If you notice during the incubation phase, we have a very low number of microbes. Okay, this is when the microbes have first gotten into the body, and we have pretty much no real immune response because the microbes aren't growing yet. During this incubation phase, you've been infected, but you don't actually feel sick. You don't know that anything's going on. The next phase is the prodromal. You'll notice that your microbes have started to increase in number, so they're replicating and dividing within the body. But our level of immune response is still pretty low. So at this point, the microbes are getting in there and growing. They've gotten past some of those first line innate systems, but we haven't started to mount a good immune response yet. During the prodromal phase, this is when you might feel some general symptoms, things that aren't associated with any particular disease, but you might be tired and achy, that kind of general, I feel sick coming on. And that's what's happening in the prodromal phase. During the illness phase, we see a huge spike in the number of microbes. They've been growing exponentially in the body now. They have well overwhelmed the tissues, and it's during this illness phase that you feel the signs and symptoms of the specific disease. And so if this is something like cholera, which is a gastrointestinal issue, you're now starting to feel the symptoms of that. If this is the flu, you're now really running a fever and you've got those real strong aches and you feel like you have the flu. And you'll notice our immune response has increased some, but it's still much lower than the microbes. This is why you are ill. This is why you feel sick, is because right now the bacteria or the microbe is winning the fight against the immune system. But eventually, in acute infections, you get better. So you start to clear those microbes. Notice the number of microbes now are going down in response to our immune system going up. That immune system has gone up very high, because remember, our innate immune system has now activated our adaptive response. It's gone through clonal selection, and we've got a lot of cells. Now the immune system is winning. We're in the decline phase, and this is when you're getting better. If you notice the number of microbes that are still in the body, though, typically you are still infectious during the decline phase of a lot of diseases. And in the convalescent phase, you feel perfectly fine. You might still have a few microbes in the body. We still have a nice, strong immune system response. And then long-term, microbes are pretty much gone. And we have a nice memory response in case we see that pathogen again. If we're going to study what makes a pathogen, first we have to be able to identify what pathogen is causing the disease. We do this using something called Koch's postulates. So Robert Koch was a scientist back in the 1800s, and he first used these postulates to identify the causative agent of anthrax. So in 1884, he identified Bacillus anthracis as the causative agent for the disease anthrax. And that was the first time anyone had connected one single species of microbe to causing one particular disease. And if you want to understand and study microbial pathogenesis, you need to be able to first identify what microbial species you should be looking at for a certain disease. With something like COVID-19, this had to be done when people started getting ill with respiratory symptoms that weren't testing positive for any of our known respiratory pathogens. This was going to be a new disease, and we had to go through things like Koch's postulates to identify what the actual pathogen was that was causing that disease. So here are what Koch's postulates say. The first is that the suspected agent must be absent in healthy organisms, but present in diseased organisms. So you can see the little red circles here is our pathogen, or what we think is our pathogen, but it's absent here. So there's nothing here in our healthy organism, but in our poor little diseased mouse here, we can see this organism. So that's a sign that maybe this microbe is what's causing the disease. To identify if it truly is, the next step is we have to get what's called a pure culture. We've talked about culturing already. It's important to get this specific microbe growing individually on the plate so that we can know we're studying the specific microbe we thought was involved. Obviously, there's no microbe in the healthy ones, so there's nothing growing on those plates. Once we grow the pure culture, then the next step is to take that microbe and inject it into a model organism. So to inject it into a healthy organism, and if this was our pathogen, then our healthy organism should get the disease like our original had, and it should have similar symptoms. The very last step is to reconfirm that we actually have that same pathogen there. We injected it in. If we get it out and the organism got sick, then that's a good sign that this pathogen or this microbe is the actual pathogen of whatever this disease is. So pathogenicity, the ability of a microbe to be a pathogen or to cause disease, is really a combination of two main factors: the infectivity of the organism and the virulence of the organism. So the infectivity measures how easily an organism actually causes disease. So how easy is it to infect you? The virulence is measuring how severe is the disease that it caused. So how deadly is it? How bad are the symptoms of the disease itself? We're going to look on the next slide. You can measure infectivity with something called the ID50 and virulence with something called the LD50. Now, pathogenicity also has other things to it. Invasion and invasiveness are two, where invasion is the ability of a pathogen to enter your cells, so to get into the cell's cytoplasm, and invasiveness is its ability to spread through tissues. But these are minor, and we're really going to focus on infectivity and virulence throughout this lecture. Okay, so if we want to study the infectivity or the virulence of an organism, we need to be able to study the ID50, which stands for the infectious dose 50, or the LD50, which stands for the lethal dose 50. And what these are looking at is how many organisms do you need to actually cause an infection, or how many organisms do you need to cause death. These are typically done in animal models, so you're infecting mice with known amounts of the pathogen and then studying to see the response. So here's what an infectious dose curve looks like. You can see down here at the bottom we have the dose of the listeria, so 10 cells, 20 cells, 50 cells, so on and so forth that were given to the mice. And here on the y-axis we have the percentage of the mice that got sick. So if you gave them 10 microbes that they ingested, just under 20% of them got sick. And what the ID50 is, so the 50 stands for the 50%, it's going to be the amount of microbes that will make 50% of the organism sick. So you can see here we get about 1,300 microbes are going to make 50% of those organisms sick. What this tells you is this is about an intermediate infectivity. So there are some pathogens that are highly infectious. Their ID50s are down in the 10 range. You also have other pathogens that are really not very infectious at all, and their ID50s are up in the 1 million range. So this is kind of intermediate. It's infectious, but it's not super infectious. You can do the same thing looking at the virulence of the organism. So again, we have the dose of listeria here, but now instead of looking at the percent of animals that were getting sick, we're looking at the mortality or the death of those organisms. And again, the LD50 is how many microbes do you need to have that leads to the death of 50% of the population. So we can come over and we come here and we find out that about 63,000. So that's a big difference. It only takes about 1,300 bacteria to make you ill, but you'd have to consume 63,000 of them to actually get a bad enough version of the disease to die from it. This tells you that this pathogen has relatively low virulence. It might cause disease, but that disease is not super fatal. When we look at pathogens, they can fall on all different ranges. So you can have pathogens that have low infectivity and high virulence. You can have ones that have low virulence and high infectivity, and vice versa. So we know some microbes can be pathogens and other microbes are not. The difference between them are called virulence factors. So they're factors, typically proteins or carbohydrates or lipids, that one microbe has that allows it to actually be a pathogen, that allows it to get into the human body, evade the immune system, so not get killed right away, and grow inside and cause damage. It's interesting when you look at microbial pathogens because these virulence factors can be as simple as one specific protein. For example, E. coli is a normal gut microbe. It's not a pathogen. It lives in all of your intestinal tracts. But a specific strain of E. coli 0157H7, is a foodborne pathogen. The difference between regular old E. coli and 0157H7 are the virulence factors that 0157H7 has. And that's just a toxin. It has an extra toxin it produces, and that toxin causes disease. So virulence factors are all the things that let a pathogen be a pathogen. When we look at them in a genome, they're actually often clustered together in one region, and we call those regions pathogenicity islands. So parts of the gene that often were acquired through horizontal gene transfer, that probably recombined into the genome and brought these extra bonus pieces of DNA for the bacteria or the virus. And now these are going to increase the ability of that microbe to grow in the human body, making them virulence factors. So the whole idea with infectious diseases is that this is really a fight between the invading microbe and the host. We already learned about the immune system and all the things that the immune system can do. So when you look at the host side here in green, we've got what their innate immune defenses are, their adaptive immune defenses, any prior immunity, so have they been vaccinated or have they seen it? We also have their health status because things like stress or increased age can weaken your immune system. And all of those factors are really based on the host genetics. So if all of this wins and the immune system wins, we get health, so we don't get disease. However, if on the pathogen side they win, and we've got a microbe that has a number of different virulence factors, maybe it has a low infectivity number, so the infectious dose is very low and the number of microbes needed to cause disease is low. Those are controlled by things like the genetic factors of the microbe, the type of pathogen it is. So there are some pathogens that are called opportunistic. They can only cause disease in people that have a weakened immune system, and kind of how the pathogen gets in. If all of the factors on the pathogen side win out in the battle, then we get disease. And so this is also an ongoing battle between the microbe coming in and the host immune system trying to fight back.